Okay, this is now continuing with session 3, where we show the data sweep function. So we have finished in the previous session, we have finished the ecological niche modeling workflow with the EGI resources, and you see here the results. The results log, you can browse them with a quick jump here. You can visualize the results by clicking the links as the outputs. Here you have the projections from the experiments. Okay, now this is the result of the ecological niche modeling workflow for that particular species. The results give you geographical information on where the suitable habitat of this species exists under different climate scenarios. Now there is a special function that is called data sweep, and if you press that, you can repeat the very same workflow under the very same conditions. That means the conditions that we have defined with all the parameter settings, the same layers, the same algorithm, the same environmental data, and you can repeat it with a different input file. So we have chosen the input file that we've chosen in the original run was this one, so we can run it again under this one. That's one species, but we can repeat the same workflow with the same parameter settings for other species as well. And these are species that you would choose which comply to the same environmental conditions as well as the same geographical conditions that we defined in the workflow. So here we have another species, and you can add as many iterations as you want. That saves you a lot of time, because this makes it possible now to run very large experiments with, let's say, 50 species that all live in the same environment, and you can now run these models, build the models and project them for 50 species at the same time. So if we're initiating this run, we can do it for 2 species now, just for the demo purpose. The sweep will start. This one, actually, the third one was an empty one, so there's no species in there. And you see the sweep is initiated, and this, you have a pending state, and then this will successively move to initializing and then finishing. This one is failed because it was empty, but the other ones, they should be initialized. So we can look at the run that I just did a couple of minutes ago. Here, 29 minutes ago, I did a sweep with these 2 species, and here you see it was finished after 4 minutes. And you have species 1 and species 2. You can look at each run individually, but you can also overview the results for all runs. So you have a matrix of each output structured according to across each species. So if you want to look at the links of the projections that resulted from the workflow, you can now compare those 2, positive link, and then you have those 2. So you can now compare. This is one species, present distribution. You see that one has quite a lot of presence in North Europe, and that's the area where, under present scenario, it's also a very high habitat suitability. And the other species, this one, that is not so present in Northern Europe, it's more a Central European one. So you can now upscale this, and let's do it for 20 or 50 species. That's one of the results we're getting from some of our investigations here. You can now produce these maps for many species. This is a marine one, by using the data sweep function. and then you can analyze these things all together. Okay, so that was the data sweep function, and of course you can download all the results. You can manage the sweep. Again, you can start sharing it, allowing other collaborators and projects to be a part of it. So, for example, BioVeL group, everyone in the BioVeL group should be able to view and edit the workflow. You can see that here. So that's the permission. So this one is a public workflow for the entire group now. So it's accessible to everyone, and everyone can see it under this run. I can't see it, but everybody else can see it under these runs. So this was the data sweep demo, and that's the end of session number 3.